A dark sphere tightens beneath the surface as thousands of small fish turn almost simultaneously. Predators circle the living mass, cutting through its edges and forcing it into denser shapes. The formation is called a bait ball, but that name describes appearance rather than intention. No fish volunteers to become bait, and the ball has no planner directing its geometry. It emerges from individual escape rules operating under concentrated attack. The apparent purpose is therefore paradoxical. Crowding attracts attention while reducing some individual risks. You can understand the sphere only by following pressure from predator to neighbor to moving boundary. Each strike changes the group's shape, and each adjustment creates new opportunities and dangers. The central investigation asks why prey compress instead of scattering across open water. Direct observation shows coordinated schooling, predator attacks, fragmentation, and repeated regrouping. Mainstream interpretation emphasizes dilution, confusion, collective detection, and limited safe space near the surface. These benefits are conditional because skilled predators can exploit predictable group behavior. A bait ball is neither perfect defense nor cooperative sacrifice, but an unstable survival response. Its rounded form records the directions from which predators and physical boundaries restrict escape. The mystery unfolds as the attackers begin herding a school that was previously moving freely. Many small pelagic fishes form schools even before an attack. Individuals align direction, maintain spacing, and respond quickly to nearby movements. Vision and the lateral line help detect neighbors, pressure changes, and approaching disturbances. Schooling can improve predator detection because many animals sample the surrounding water. When one fish turns sharply, nearby fish respond, spreading an escape wave through the group. Under attack, individuals tend to move away from predators and toward neighboring fish. Those simple reactions reduce isolation but compress the school as threats approach from several sides. The surface can become a ceiling, preventing upward escape and flattening or rounding the aggregation. Predators below and around the school further shrink the available safe volume. The result may be a rotating, pulsating ball whose boundary changes with every strike. There is no fixed membership because fish exchange positions as the group turns. Individuals near the edge face direct attack, but may also find open routes away. Fish within the center gain dilution, but encounter crowding, reduced oxygen exchange, and limited maneuvering. The dilution effect means any one fish has a lower chance of capture when surrounded by many alternatives. Confusion can reduce a predator's ability to track one target among crossing, flashing bodies. This effect depends on predator sensory capacity, attack speed, and the group's visual density. Predators can counter confusion by isolating individuals or attacking a narrow portion of the boundary. Coordinated hunters may divide roles, with some compressing prey while others strike. Humpback whales, Megaptera novae, sometimes use bubbles and coordinated movement to concentrate schooling prey. Common dolphins, Delphinus delphis, can herd small fishes into compact surface aggregations. Seabirds attack from above, adding another direction of pressure and closing possible exits. Sharks, tunas, and other predatory fishes may strike through or along the school. Different predator combinations produce different bait ball shapes, durations, and escape opportunities. The first explanation to test is that prey deliberately form a ball to confuse attackers. Confusion is supported by experiments and observations of reduced tracking efficiency in some group hunting situations. However, the ball also arises because escape routes are constrained, not because fish choose an ideal sculpture. The second explanation is simple selfish crowding, where each fish seeks neighbors to reduce personal risk. Movement toward the group can create a compact aggregate without communication about collective goals. This selfish herd model explains compression, but does not capture every alignment and information sharing benefit. A third explanation claims predators intentionally preserve the ball as a renewable feeding resource. Predators do herd prey, yet observed attacks often disperse, consume, or exhaust the aggregation. No general evidence supports a shared agreement to maintain prey beyond immediate hunting advantage. Another misconception treats synchronized turns as proof of telepathy or a single group mind. Local sensory responses can spread faster than a viewer can distinguish individual decisions. When a predator charges, edgefish accelerate and turn away from the incoming path. Neighbors match that movement. The turn propagates, and the entire boundary folds inward. The adaptation preserves cohesion, while the consequence is greater density elsewhere in the ball. A predator then targets the compressed edge, forcing another wave through the school. Repeated waves create shimmering bands that reveal alignment, body orientation, and reflected light. If the attacker separates one fish, confusion falls and capture becomes easier. Prey, therefore, rush back toward companions unless an open water escape offers better odds. This cue action sequence continually rebuilds the very structure predators are trying to exploit. The ball may split when an attacker passes through its center. Fragments can recombine after the strike, or flee in different directions if pressure briefly weakens. You might see coordinated resilience, but the outcome can still be devastating for many individuals. Duration ranges from brief compression to prolonged pursuit, depending on prey, predators, and local conditions. Observations from one spectacular event cannot define a universal strategy across all schooling species. Measurements are complicated because boats, divers, and cameras may change predator or prey behavior. The strongest conclusion is conditional. Aggregation redistributes risk, while predators adapt to its weaknesses. Bait balls connect individual decisions to energy transfer across entire marine food webs. Small fishes often concentrate energy gathered from plankton into packages large predators can capture. When herding succeeds, that energy becomes available to dolphins, whales, sharks, tunas, and seabirds. Prey fish sense an approaching body and compress toward neighbors before direct contact. Their rapid turns preserve the school, but crowding creates a dense target at the surface. Seabirds then dive from above, converting an aquatic refuge into a vertically enclosed trap. Fishes respond by plunging or splitting, and aquatic predators redirect the fragments upward again. This alternating pressure can cycle until prey escape, disperse, or are consumed. The event may feed scavengers as injured fish, and scraps descend below the main hunt. Nutrients move through predators and waste, connecting the surface spectacle to deeper communities. For humans, bait balls can indicate feeding opportunities, but do not automatically measure ecosystem health. A large aggregation may reflect abundant prey, intense predation, unusual currents, or temporary compression. Fishing near such events can add pressure at moments when prey and predators are already concentrated. Removing forage fishes at broad scales can affect predators, fisheries, and ecosystem resilience. Management therefore considers total populations and food web roles, not isolated dramatic gatherings. Ocean warming can shift plankton, forage fish distributions, and the overlap between hunters and prey. Deoxygenation may compress suitable habitat vertically, increasing encounters in a thinner surface layer. Changes in water clarity can alter detection ranges and the effectiveness of confusion. Underwater noise may influence some predator-prey interactions, though effects vary and should not be generalized. The bait ball becomes a visible intersection of behavior, environment, and rapidly transferred energy. Its secret purpose belongs to individual survival, while its ecological consequence extends far beyond the school. Future research needs three-dimensional tracking of prey and predators throughout complete natural encounters. Individual trajectories can test whether fish seek the center, match neighbors, or exploit temporary gaps. Simultaneous recordings above and below water can reveal how seabirds change underwater escape geometry. Repeated studies should compare predator combinations, rather than treating every bait ball as equivalent. Researchers can measure capture rates before, during, and after compression to test defensive benefits. They should also record failed hunts, because spectacular feeding footage overrepresents successful predators. Models can recreate local movement rules, but agreement with shape alone does not prove the rules are correct. Predictions must match splitting, regrouping, edge exchange, and individual survival under real attacks. Environmental measurements can test how oxygen, light, currents, and depth constrain available escape space. You would still not know a fish's intention from its position inside the sphere. What science can establish is how repeated local responses generate measurable group-level consequences. Unknowns remain about decision thresholds, fatigue, learning, and differences among individual prey. Predators also change tactics, making the bait ball a moving contest rather than a stable adaptation. When the dark sphere reappears, its boundary will again be written by every approaching hunter. The bait ball's scientifically supported purpose is conditional defense, born from individual escape and continually reshaped by predation.